Good morning, everyone. My name is Jessica Page. I'm a minor in biology, and this summer my advisor was Dr. Schuerger here, and my project was entitled The Survival and Growth of Spacecraft Microorganisms Under Simulated Martian Conditions. So a little bit of background. Serratio liquefaciens has already been known to survive the void of space and Martian conditions by experiments done by Dr. Schuerger. And so what I'm doing is further investigating this phenomena by looking at the survival and growth, including the radiation, the other conditions, and the effect of soil that could possibly be covering the ice. So why is this important? Well, there have been life detection experiments on Mars. This is a picture of Viking, and then we have rovers and such on Mars. So we want to make sure that we're not contaminating Mars with the bacteria from Earth, and yes, we don't want to contaminate the life detection experiments that work. So the experimental conditions on Mars that I worked with, the pressure is 7 millibars, which is less than 1% of Earth pressure. The temperature I used was minus 25 to 20 degrees Celsius, and I'll explain why I use that specific range in a moment. The atmosphere on Mars is mostly carbon dioxide, 95% carbon dioxide. A little bit of nitrogen, argon, oxygen, tiny bit of water. And there's a much higher influx of UV radiation onto Mars than there is on Earth, and that's a really key factor that's going to come. Growth and survival in, or sterile deionized water, phosphate buffer, and 5 and 10% compositions of magnesium sulfate, because those are found in the Martian regolith. So we were looking at which type of concentration was better for survival and growth. And then the phosphate buffer is kind of like a positive control in that phosphate should aid growth, so we would want to see more growth and survival in those solutions, and we'll look at the results from that. Here, it has the full range of UV light from 200 to 400 nanometers. UVA, UVB, and UVC. UVC is actually not found on Earth. It is the shortest wavelength, highest energy, the most biocidal, and it doesn't reach Earth's surface because our ozone layer protects us from it. So it's another reason why it's very important to protect the ozone layer, because it is the most biocidal. So the Martian simulation chamber used all of these wavelengths that we have here. Liquefaciens, this is a picture of a culture of it on TSA. It's a cyclophilic hypobarophile, which means it's cold tolerant, it's mildly tolerant to salty environments, and it's tolerant to low pressure. It actually grows pretty well in low pressure, as we'll see. It's gram-negative, rod-shaped, and a facultative anaerobe, meaning it can survive with or without oxygen. An overview of the approach to this project. I started with just looking at the effects of UV, not Martian conditions, in chamber to determine the freezing temperatures of the salts that I was testing the growth in. And that was also a process of just getting to know the Martian simulation chamber and getting familiar with working with it. And then moved on to the effect of ice versus liquid in UV shielding, because we wanted to see if being frozen would shield it from the UV light or not, and I'll go into that. And then finally adding in the UV light plus the Martian conditions, and then adding in the Martian soil with that. Day experiments, what we're using, this is the Martian simulation chamber built by Dr. Schuerger. It simulates the pressure and the temperature and the atmospheric content of Mars. So I would put the samples in here and run it. And then this is the laminar flow head where I processed all the bacteria after exposures and such. And this is for the UV experiments only, this was within the Mars chamber. This is a magnet and a magnetic stirring rod, and the solution with to do with the results that I'm going to show in a moment. So when I talk about the stir bar, this is what I'm talking about. We think that it could be shielding the bacteria from the UV light. Maybe some of the colonies are getting under the stir bar and shielding from the UV light, because we did see some survival, as you will see. So for that portion of the experiment with just the UV light, We tested UV exposure times from 0 to an hour in these increments here. Just Earth conditions, as I said, and only looking at solutions to look at the difference in survival. So here are the results from that. As you can see, after just 10 seconds of UV exposure, we went down 3 orders of magnitude in bacterial survival. And then even more so after 30 seconds, and then you can see it kind of levels off all the way out to an hour of exposure, which you would think, based on the initial results, that we wouldn't see any survival at all. So we think that it could be due to the magnetic stir bar shielding from the UV light. And that we have, if we have surviving bacteria on a rover or something on Mars, and it's shielded from the UV light under a wheel or a tire or something like that, we want to make sure that it's not shielded, that it doesn't survive. So the next phase was, like I said, working with the Martian simulation chamber, getting to know exactly how to work it, optimizing the experimental protocols. So I determined the freezing points. They were all determined to be between 5, including the soil. The Martian soil in the solution had no effect on the freezing. And I found that I also had to account for a supersaturated layer in the magnesium sulfate solutions, because it would take longer to freeze. So that's why I had to go down to -25. That wasn't the freezing temperature, but the conditions were tested in -25 degrees Celsius because of that. And I have a picture of that. You see like this white layer, that took a lot longer of a time to freeze in the solutions, and that was a factor. Another thing I noticed, or we noticed, is when we were repressuring after we had exposed to the UV light in the Mars chamber, the salts would splatter everywhere in the chamber, which is could be potentially dangerous racial liquefations. It could be a pathogen. It's not that dangerous, but it is a little bit. But I mean, this is also a concern because we were noticing that the salts, when they were coming back up to liquid from while on Mars, in a Mars situation. So then this was pretty much the final phase that I have data for putting everything together. We have the Martian conditions because we're in the Mars chamber, the liquid solutions that are about to be frozen. These are going to be exposed to the UV light because the UV light is coming down from up there, and we're exposing the cells right under there. And then the exact same 4 solutions are back here under this aluminum UV light to compare the effects of those two. So here are the results from that. As you can see, okay, so blue is added UV exposure. In the blue, they were exposed to the UV. In the red, they were in the Mars chamber, but not exposed to UV. So they were still undergoing the temperature and the pressure changes in the atmospheric content. And then the green is just Earth normal conditions, plated in the plates immediately, no UV salt solutions we tested. You see the same trend in everyone, and this, by the way, is without shielding from the magnetic stir bar. So this is just directly exposed, no stirring. So as you can see, after about a minute or so, or 10 seconds even, you get almost no survival. So it's definitely the UV light that is the most biocidal effect in these experiments. Make sure there's nothing else to say about this. And then the survival with just the Martian conditions, a little bit, not as significant as the added UV exposure, but the Martian conditions are a little bit biocidal, the temperature and the pressure. So right now I'm just testing the effects of the salt solutions themselves to make sure that they're not adding to the survival or death of the cells, as well as them being frozen, so freezing them for 24 hours and looking at that. So far, I don't have enough data to generate a graph from it yet, but so far it looks like actually in the liquid solutions, the survival increased a little bit, so it's not really affecting our biocidal effects that much. So yes, and then soil will test the effect of Martian soil, because Martian soil can also shield, could possibly shield the bacteria from the UV light. So, yeah, in conclusion, the UV light is very bactericidal under Martian conditions, so we should consider making sure that they are not shielded from the UV light. And yeah, I also just wanted to thank Space Florida and NASA's Florida Space Grant Consortium, Dr. Schuerger, everybody here, Brian, everybody at the Space Life Sciences Lab. Thank you guys very much. This was an awesome opportunity and had a really great time this summer. So. Any questions?
So the guys that did survive, did you try to grow them up and see?
Yes, actually I did, and I did have a slide, but I didn't put it in the presentation. But yes, non-diluted. I didn't dilute them at all. I immediately plated them right after that, and there was survival. There was definitely. I can't think of the numbers right now. It really depended. For the phosphate buffer, for instance, the entire TSA plate was filled with colonies, and then.
Did they look different, or have any morphology differences, or growth rate differences?
Not really. They didn't. The morphologies didn't really look that different.
And then did you take those guys and repeat the experiment to see if more would survive with your first survivor? Meaning do a second generation of the experiment from just
with the same colonies?
With your population of survivors?
With the same colonies? I'm sorry, what was the question?
Yeah, the guys that survived, you said you were able to grow them up, and they looked normal.
Yes.
Physiology-wise, anyway. Did you take that population and repeat the experiment?
No, I didn't, but that would, yeah,
So
that's a good suggestion.
10% survive instead of just 1%?
Yeah, that's a good idea. I'll try that next time. Yeah. Any other questions?
If there's no more questions, I'll make just to add a point here, is that the data that Jessica generated this summer is going to be added with data that we had another student who was visiting from the University of Arkansas, and we're combining that into a paper that'll probably go to astrobiology to be submitted sometime later on this fall. We don't have a working title or anything, so we left it out, but she will get into a paper.
That's very exciting. I did not know it was an astrobiology.
Oh yeah, it's going to astrobiology. So.
Well, that's one thing, it's great that you do this internship, but that the fact that you're all getting a paper out of it or a poster paper out of it, that's amazing. Because I see a lot of internship where they sit and do the work, they go back and forget about it. So this is great.
Yes, we're very grateful.
Okay, thanks.
Jessica?
Ready? 1,